Chapter 17, Mutations. Before we talk about mutations, we should go over the steps of gene expression. Gene expression is a two-step process wherein a gene is copied, RNA is made, and RNA is read by ribosome and in codons, which is 3 nucleotides at a time, and tRNA, which match up with the codons, with their anticodons, complementary anticodons, carry the amino acid to the ribosome, and ribosome is going to take those amino acids and make peptide bonds between them. So this chain of amino acids is made, which is a protein. This is how we make use of genes and make proteins. This is called gene expression. So if the gene changes, then the copy of the gene, which is RNA, also will change. And when copy of the gene has changed, then the kind of amino acids that make the protein will be changed because different tRNAs are going to read those codons. This is how the mutations in gene, which are nothing but changes in the nucleotides of a gene sequence have changed, will impact our gene expression. If this is a gene for insulin, if it is mutated, then we would make a faulty insulin. And this can happen with any gene. Some mutations are okay, we can still survive, but other mutations are lethal, so organisms may not survive. Coming to definition of coming to the definition of mutations. Mutation means change in the genetic material. These mutations are spontaneous when they occur during the DNA replication, or at the time of repair, or during the time of crossing over and recombination. And these mutations are occurring because of the changes in the environment. If a organism is exposed to harmful rays such as X-rays and or UV rays, then the mutations can happen in the DNA. And sometimes chemicals also can cause changes in DNA, and some of these chemicals which result in mutations which cause cancer, then they are called carcinogens. So not every mutagen is a carcinogen. Mutations are of two different types. If they are in large scale in chromosome, then they are called chromosomal mutations. That was covered in chapter 15. And if the mutations occur due to single base pair change, just one base pair has changed, then it is called point mutation. So as you can see, this is a gene for hemoglobin, and just because this T was replaced by A, now the gene of mutated hemoglobin has CAT in its gene, which will be copied during translation into GUA instead of GAA. And the tRNA reads GAA and adds amino acid glue, whereas GUA will code for amino acid valine, and replacement of this amino acid by a different amino acid called valine results in a hemoglobin which has a different structure. And because of the different structure, hemoglobins, which are the protein molecules in red blood cells, they start to polymerize when there is less oxygen in their red blood cells. And because of that, they form crystals. And when the red blood cells form crystals, red blood cells lose their shape and they become sickle cell shaped because because of that there will be interruption in the blood flow, and because of that many other consequences result, and it results in sometimes even in death. So there are different types of point mutations. They're all categorized into two. Um, One is called substitution mutation, nucleotide pair substitution mutation, and second category is called insertion or deletion mutation. So let's see, what is substitution mutation? One nucleotide is replaced by another nucleotide, as simple as that. If this replacement occurs in between genes, then it will not have any impact pretty much on the organism. Then it is called silent mutation. If at all it occurs in introns in eukaryotes, then again it will not have any effect. Then such mutations are called silent mutations. If a mutation occurs and the nucleotide change has resulted in the placement of a wrong amino acid, for example, sickle cell anemia, right? Sickle cell hemoglobin. Just because one amino acid has been replaced by another amino acid, it resulted in a different a nucleotide replaced by another nucleotide, it resulted in a different amino acid, and that is called missense substitution mutation. Coming to nonsense mutation. Sometimes what happens is, because of this replacement of nucleotide in the gene, the RNA is going to be read by ribosome, but when it is reading it, it suddenly encounters a stop codon. So that replacement has resulted in the formation of a stop codon in the middle of the gene. So this results in a abrupt stopping of translation or a non-functional protein. Such mutation is called nonsense substitution mutation. So remember, point mutations are of two types: substitution and insertion or deletion mutations. Substitution mutations are further divided into three types: silent, missense, nonsense mutation. The point to remember: sickle cell anemia is which kind of mutation? It's a point mutation. It is a substitution mutation. It is a missense type of substitution mutation. Point to remember. These are all the examples of these mutations. Coming to insertion or deletion mutations. Because of addition of a nucleotide, the frame has been shifted. Remember, during translation, 3 nucleotides are read at a time in an RNA by ribosome. So if a nucleotide has been added and it results in a changed protein or abrupt stopping of translation, it could be anything. Just because of one nucleotide extra in the RNA, anything can happen. So frame has been shifted. So nucleotides which are read 3 at a time, the frame has been shifted. Hence, the insertion mutation is called frameshift mutation. Same is the case with deletion mutation. Here there should have been a U, but because A was missing, there is no U here in RNA. Because of that, UUG will be read instead of reading UUU. So instead of placing phenylalanine in the chain of amino acids, this protein would have leucine, and this would change the entire protein. Remember, primary structure of the protein is very important. We read this in the structure and function of organic molecules chapter. So addition or deletion mutations, they change the reading frame. That is why they are called frameshift mutations. So this is frameshift mutations or point mutations. these are some of the questions that you can see in the test. If this is the template, what will be the non-template? All you have to do is for 3 prime, there will be 5 prime, T to A, C to G, A to T, like that. You have to make DNA first. And for mRNA, you have to use the template sequence. Do not use the non-template sequence. Use the template sequence only. So you have to start from 5 prime, and for mRNA, so for 3 prime it will be 5 prime, and you start from here. So this will be the mRNA, T to A, C to G, A to U, because this is mRNA, so on and so forth. So always remember, template should be used for transcription, that is using DNA, one strand of the DNA, and making mRNA sequence, which is complementary to the template strand only. In test questions, they specify which one is the template strand. By looking at a sequence, nobody can determine which one is template. Once you have the mRNA sequence, you have to start reading from 5 prime and 3 nucleotides onwards, and you have to start at AUG. If they haven't provided anything and they asked you to simply translate it, then they will be giving you the codon table, and you have to use 3 nucleotides, match these UGC in the codon table, and you will see cysteine amino acid, and simply do this translation. So once UGA is seen, you have to simply say stop. That is where translation stops. It does not continue like this. There is codon in RNA and anticodon in tRNA. There's codon in mRNA and complementary anticodon on tRNA. tRNA has anticodon and a specific amino acid on its amino acid binding site. So for GCA, complementary would be CGU. Remember, they're tRNA. So it will place, tRNA will bring in alanine to ribosome. It will not be complementary to GCG. So tRNA, when it comes to the ribosome, by swimming around, it does not walk to it voluntarily, and it will just bump into it, and if it bumps into GCG, it will not even recognize. These are some of the questions which give you more practice for using template strand, and it's always start at 5 prime end for mRNA sequence, and read 3 nucleotides at a time from mRNA for translation. And for this you need codon table AUG. When you see in codon table, it always codes for met amino acid, and AAC, it codes for ASN amino acid, so on and so forth. This is one of the questions wherein they have given you the mutated DNA sequence. As you can see, the template strand is the top one. So you made the sequence, and the amino acid sequence will be this, and at the very end it is a stop. So this is, instead of met, ASN, ARG, leucine, and stop, what will you get? That will be the answer in the test. Okay, practice some of these questions. I have seen this kind of question last time, and also this kind of question several times in final exams and competitive tests. This is a definite question